Okay, just to clarify some aspects, and I probably confused people before as well, so I made sure this was clarified in class. I just wanted to go over something about the carotid bodies and the chemoreceptors and the central nervous system also. So it's really interesting. I read this paper and I put it into files under Canvas. It's this whole symposium that was all on the carotid bodies. It was really quite interesting, in the sense that this guy, De Castro, actually described the carotid bodies and the carotid sinuses back in the early 1900s. So in 1926, here's a nice figure that he drew and stained up the carotid bodies and all this work that happened from here. So he was really the first one to do a lot of the anatomy. And what we see here are these original cells of the carotid body, and they call these glomerulus or glomus cells. But these cells here, you can see them over here, and then these are neurons. These are the sensor neurons that come around these cells. All right, and then he also did a lot of anatomy on the carotid sinuses that detect pressure, the baroreceptors, and their fine endings. And here they are, he's stained up and drawn. Oh, this is pretty interesting. And there's De Castro. So these, they also call these type 1 cells, neurons. So what's interesting is that these cells here, these type 1 cells they call them, are actually the sensors for pH, CO2, and actually thought to be responsible for monitoring glucose now. They don't know if it can detect the body's glucose and has an effect in the sense of altering the neural function, but that's interesting. Anyways, this guy De Castro described all this back in the 1920s, and he related to the guy named Heymans in Belgium, I think it was. Yeah, it might have been Belgium or the Netherlands. Anyways, and Heymans was put up for the Nobel Prize like 3 times, and then finally got it in 1938 in physiology and medicine. But it was actually from the work of De Castro. It's kind of interesting in this review that if you wanted to read the intro chapter, it says that the Nobel Committee wasn't sure if De Castro was alive to give him the prize because the Spanish War was going on, and a lot of people had died during the Spanish War. And so he probably would have got the Nobel Prize because he was the one that really described the anatomy really well and actually shared it with Heymans' work. So it's kind of interesting to think about how the politics has impacted this guy not getting the Nobel Prize. Anyways, with all the work in people looking at this, these type 1 cells that respond to the stimulus, they also have some other names, but type 2 cells are more supportive cells in this region, like I guess some of these supportive cells around here. So type 1 cells, they detect the low pH, but they also detect low partial pressure of oxygen. Now, there's still a debate, does it really detect the partial pressure of oxygen or oxygen content? So that's still debated even to this day, it seems like. But the thing is, they are very sensitive to low oxygen, maybe more sensitive to low oxygen than they are low pH. So I think I have confused people with that oxygen saturation finger thing that we use, the percent saturation in our little experiment that we use, because your PO2 doesn't drop that much. Well, that oxygen saturation on your fingertip doesn't pick it up as well as your carotid body, supposedly. So it's actually better to think about these cells are very sensitive to oxygen. They can also respond to pH, but I'll talk about that in a minute. These cells then, after they're stimulated by these factors, they can release dopamine, acetylcholine, ATP, norepinephrine, hypoxic factor as one, and erythropoietin. And some studies now think that erythropoietin, it's actually released in the sense of this low oxygen, and then it goes to the bone to make red blood cells. And there's a debate about if it's releasing enough to actually have an impact on red cell production, but they do know that it's highly concentrated in these cells. That's kind of interesting. So these cells release these substances like dopamine, acetylcholine, ATP, norepinephrine, and they bind to the receptors on the nerve endings and then cause the nerve endings to fire. So the nerve endings are not picking up pH or PO2. They are picking up what is released from these type 1 cells. And they now know that these neurons have all these different receptors on them, and they're still doing the pharmacology on them. So it's kind of interesting if one neuron's responding to both of these, or the different neurons respond in different ways. So they do these nice physiological recordings, some pretty cool recordings where they're actually recording in the type 1 cells, and they show hypoxia here depolarizes as soon as they give it hypoxia. And if they record from the nerve now, then they see that the nerve fires when it's exposed to hypoxia from the cells around it, releasing whatever transmitter. And people have blocked transmission release and see that these nerves don't fire. So these are the sensory nerves. So here's something interesting. They give hypoxia. This is a recording of the nerves down here. They give hypoxia at a standard CO2 level and keep the pH the same. So this is what's called isohydric hypercapnia. So it's higher CO2, but the pH hasn't changed. They keep that constant isohydric. And it responds to the hypoxia. And even with 10% CO2, it will respond. And this is the idea that even low glucose levels here, like 0.1 millimolar glucose, that it can respond to glucose. So it might be a glucose monitoring, and people are wondering, then does it really have that much physiological function, these type 1 cells, that altering this, the neuronal firing, what would be the rationale for this, for controlling respiration? But anyways, they note that they do respond to the glucose. And then, where these are in the cell bodies for the sensory nerves, going up into the 9th cranial nerve, and it goes in here and then up into the brain. Okay, but here's a blow-up of this carotid body, which is really quite interesting. Here's a type 1 glomerulus cells, and you see the sensory endings, but you also see sympathetic neurons going to this region, and there's still information to try to figure out what does the sympathetic system do to these cells inside of the carotid body. And it seems to be a question that's still bugging people, and this is what people are still trying to study. So this is really interesting. There's more to it than what meets the eye. Okay, and so what's happening with this percent saturation and oxygen? Well, we see here your percent saturation changes a little bit. Let's say it's 10 right here, but your oxygen concentration in your body, this would be like arterial oxygen, it's changed a lot from 100 to 60, and it's only changed a tiny bit here on the percent saturation. So the point is that we're having much greater oxygen changes and low percent saturation changes, and this is picked up with the pulse ox. So the pulse ox isn't as sensitive as our carotid bodies are for the change in oxygen. And so that's kind of interesting too. And then there were generalities about the saturation, like from a pulse ox. If you're watching a pulse ox, 100 to 90, it's kind of like, hey, I got to watch out for this low end over here. But that means that could be in the range from 100 to 60 millimeters of arterial, this is PA, small a, oxygen content. So, or partial pressure of oxygen in the arterial bed. So there's a large drop in the range that this could happen for that only 10% saturation. When it drops down to 90 to 80, it could really drop the arterial oxygen quite a bit. So that's even a worse effect. And then it's really bad if it's less than 80. It could be as low as down to 40. So just by a general principle, they just divide by 2 to figure out on this low end, if it's less than 80, divide by 2, and you'll probably know the arterial oxygen. And if it's in between 90 and 80, just by 1.5 millimeter, and then for 100 to 40, every 4 millimeters of mercury for every single percent drop. So kind of interesting. Oh, not multiply, but yeah, decreases by 1.5 millimeters of mercury for every single percent oxygen drop. So that's kind of neat. I wanted to clarify that and make sure that we're on the same page. And I think that's really good. So that little experiments that we do with the pulse ox isn't good enough to think about saturation in arterial oxygen with breathing. It's not really the CO2 that's building up and pH driving the breathing. It's really the low oxygen that happens and the pulse ox can't pick up a good amount of the oxygen that's in the arterial blood. Now CO2 can drive it, but here's the point I brought up in class: aviator hypoxia. This is really interesting. I talked to Dr. Lee over in physiology. He teaches respiratory physiology, and I talked to respiratory pulmonologist at the hospital. It was kind of interesting because what they found out is this really neat experiment. That was actually the Air Force that did a lot of this work, and that was what's called aviator hypoxia. So aviator hypoxia, you go up to high altitude, and what happens? The partial pressure of oxygen drops, and your CO2 in your blood is still okay, right? You go up, so now all of a sudden you breathe this low oxygen. You will stimulate yourself to breathe faster because of the low oxygen. And then what happens to your CO2 level? Well, if you're blowing out faster, your CO2 is dropping, right? You're blowing it out. So your pH becomes basic. So here you are basic, but because of the low oxygen, you're breathing very fast. So your carotid bodies are sensitive to that oxygen, even though your blood is basic. So they call this aviator hypoxia. And that helps to show that even though your blood is basic, you're breathing really fast because of low oxygen. Now, this guy DeCastro did some neat experiments, but so did Hammonds. Hammonds did these experiments in dogs and different animals, and he made them hypoxic. And then when he cut the nerves to the carotid body, they no longer breathed any faster. So he could decrease this hypoxic drive just by cutting the nerves. And then they would go back to normal breathing rate. So it kind of shows that it's very sensitive to the hypoxia. There's been debates about that over the years because CO2 might change a little bit too if they make them hypoxic. And then you also have the aortic bodies as well. And it depends on the animal that one's testing to have the extent or the drive that one has, rabbits versus dogs and cats. There seems to be different sensitivities. All right, well, that's what I wanted to go over to make sure we're all clear. So the take home of all this is that it's mostly oxygen driven and not pH driven.